Hello everyone, my name is Anthony Bui. This is part 2 of exam 3 oral presentations. On slide 1, we'll be focused on part A, particularly on figure A as attached on the right hand side. So the objective of this first experiment is trying to verify if the ERBB2 receptor located in both the cytoplasm and nucleus, as well as evaluate if the important beta 1 protein make any change in the cytoplasm or the nucleus at the level of the ERBB2 receptor. If it does, then we can conclude that the important beta level in the cell will be modulated the cytoplasmic to the nuclear ratio at the ERBB2 level. So they trying to see if the important beta 1 is required for the nuclear localization of ERBB2 receptor. And the function of the scRNA, which is a interference RNA, they bind to the 3' untranslated region of mRNA and decreases the important beta 1 level in cells. So we see from figure A that the change in of the cytoplasmic and the nuclear ERBB2 receptor level by the immunoplan test of the ERBB2. So from the top panel of the figures, we can understand that we can see that the interference RNA decreases both the cytoplasmic level of the upper, as we see in the upper line 2. So the nuclear level of the important beta 1 is seen in upper line 5, but later completely dropped. As you can see, it completely disappear. So we also see that the cytoplasmic and the nuclear level of the important beta 1 are not really changed in both the negative and dominant negative control seen in line 1, 3, 4, and 6. Therefore, we conclude that no protein was localized in the nucleus. From the bottom panels, we understand that there was a decrease of important beta 1 level due to the decrease of the nuclear level of the ERBB2 in the bottom line 4, but it does not decrease the cytoplasmic level of the ERBB2 in the bottom line 2. So we conclude that the important beta 1 have to control the cytoplasmic and nuclear level of the ERBB2 receptors. Moving on to figure B on the right hand side. So the objective of this experiment is trying to verify if the deletion mechanism of the NLS will stand for nuclear localization signals or any other mechanism of the important beta 1 that can act on the ERBB2 receptor. So scientists evaluate if the important beta 1 have to control the cytoplasmic and nuclear level of ERBB2 or not, and it also helps in the point of the ERBB2 to the nucleus. So from this figure B, we conclude that the important beta 1 bind to the nuclear localization signal of the ERBB2 receptors, and the deletion of the NLS from the important beta 1 ERBB2 will block the interaction of the important beta 1. So the immunoplan test result indicate that the important beta 1 was not appear in the immunoprecipitate portion if we missing the NLS of ERBB2. So this supports the fact that important of ERBB2 to the nucleus. So for the third question C, what can you prove? What can you propose an experiment that can research have performed? So another experiment that the research could have performed is that they can evaluate the overexpression of only important beta 1 or only ERBB2 receptor, or even both of these protein receptors. The result from this experiment will allow scientists and researchers to study the cytoplasmic and nuclear level of ERBB2, just like the experiment we described in figure B earlier. So moving on to the last slide of part B, figure C. So by looking at the figure 16-33 on the textbook, I understand that RAN is the GTP-based enzyme that acts as the RAN GDP in the nucleus, but RAN GDP in the cytoplasm. So RAN helps to transport protein from the nucleus to cytoplasm and reversely. So the first step of this figure is how important, which is receptor bind to the NLS and mediate the movement of the NLS protein to the nuclear pore. In the cytoplasm, this transport NLS protein complex is transported to the nucleus at the center of the nuclear pore complex, which is NPC. At the nucleus, the important molecule associated with RAN, which is a TDP binding protein, as I said earlier, that cause the NLS containing protein complex to be released in the nucleus. So now we have the important RAN GDP complex being transported back through the nuclear pore to the cytosol and bind to the RAN GAP. RAN GAP is a GDP-based activin protein. So the important is then released and associated with the hydrolysis of RAN-bound GDP to form RAN GDP. So RAN GDP is translocated in a to the nucleus by the NF2 protein receptor, and then GDP is then exchanged for GDP by GEF. GEF stands for guanine nucleotide exchange factors to form the GTP. So GEF is a protein that helps to release GDP from RAS protein to acquire GTP, and RAN GTP is bound to the nuclear protein and being translocated by the important beta receptors. So on question 2, we see that on the figure C, we have 293 cell that was transfected with wild type RAN Q69 RAN mutant cells, wild type ERBB2, and the control vector negative. So since the mutant Q69L was mutant, the RAN is unable to hydrolyze GDP to GDP. Therefore, the Q69 RAN mutant is defective with GDPase, and then the cytoplasm nucleus and the total lysate fraction were isolated to perform the Western blot test with the ERBB2. So the important beta 1 and RAN, as the result, we see that the cytoplasmic fraction, the ERBB2 and important beta 1 level are not changed in the wild type Q69L mutant and control vectors. So this means that there's no change in the ERBB2 level due to the action of important beta 1 in cytoplasm. So we see the equal expression in nuclear fraction of important beta 1 and ERBB2 level in vector control in wild type cell. Therefore, we conclude that ERBB2 receptor wild type is not overexpressed in the nucleus, but the important beta 1 will bind to the ERBB2. So the control vector is believed to have normal ERBB2 and important beta level as the wild type cell. However, the mutant Q69L has a reduced level of important beta 1 and ERBB2. So we conclude that RAN GTP was mutated in the nucleus and reduced the level of ERBB2 and important beta 1. So since the mutant Q69L was defective, it cannot form RAN GTP anymore to move to the nucleus and exchange for the RAN GTP by GEF. That's for the nuclear dissociation of important beta and ERBB2 will not form, and we have the important beta 1 RAN GTP complex cannot longer make RAN GTP and important in the cytoplasm to bind new ERBB2 anymore. So the result is that we have a low level of important beta and ERBB2 in the nucleus of mutant Q69L mutant. The Western plot of lysate result indicate that they're trying to verify if RAN was present in the wild type and Q69L mutant cells. So we see that RAN is expressed in equal amount in wild type and Q69L cells. However, RAN was not transfected in the absence of control cell. Meanwhile, we have the important beta level and not changed in all three treatment of the cells. And from the model, we can propose a model that ERBB2 binds to important beta 1 and is translocated to the nucleus by NPC in the wild type cell, NPC I mentioned earlier. We expect to see the ERBB2 important beta 1 complex bind to the RAN GTP by the exchange of GTP to GDP on RAN by the GEF mechanism. The result will have the separation of ERBB2 from important beta 1. Meanwhile, we have the RAN GTP bound with important that will enter the cytoplasm and form the RAN GDP. Meanwhile, we also have the ERBB2 expected to bind to the important beta 1 in the cytoplasm of mutant cells, and both remain the same amount levels. However, when this complex gets into the nucleus, it will not bind to the RAN GDP because of the defective GTPase of RAN. So in fact, important beta 1 will bind to ERBB2, which indicate a low expression of ERBB2 and important beta 1 in the nucleus of mutant Q69L. Saying important beta 1 cannot be transported from the nucleus to cytoplasm, it will also block the nucleic translocation of ERBB2 from the cytoplasm to nucleus. Therefore, the mutant Q69L will reduce the ERBB2 and important beta 1 level in the nucleus. So now we conclude that GDPase enzyme of RAN will allow transportation of ERBB2 from cytoplasm and nucleus. And the last question asking whether if research can replace RAN Q69L to RAN GPP-NP, the answer is definitely yes, because both of these forms considered non-hydrolyzable GTP. Therefore, you are guaranteed to have the RAN that was in the RAN GTP conformation instead of using the regular RAN GTP, which is a GTPase enzyme. it will slowly convert GTP to GDP. Therefore, you will have the undefined mixture of range GTP and range GDP, and it will confuse your results. And this guy, range GPPNP, is localized only in the nucleus and binds to the important beta 1 ERBB2 complex and release ERBB2 in the nucleus. This guy, range GPPNB, cannot enter the cytoplasm, so it will not generate important. Meanwhile, the range Q69L can enter the cytoplasm but cannot from the range GDP. Therefore, the RAF GPNP will have to increase the cytoplasm level of important beta 1 and ERBB2 nucleus while reducing the level of important beta 1 and ERBB2 in the cytoplasm as we compare to the wild type because the protein are transported to the nucleus. So that's how the result vary from the conclusion and will be different.